right, everybody, welcome back to Drug Dealin'. This is episode 3, and we're really excited to bring this episode to you today because it's a very special episode in that it is our first interview with a biotech leader. You guys are probably sick of hearing just me and Kenny rattle off takes in our first couple episodes, and we're really excited to give you a little bit of a click into this nice conversation that we had with the biotech leader. We had the pleasure of having Sujal Shah join our podcast today for a long conversation about his career and the ups and downs of running a biotech company. Kenny, you want to say a few words about Sujal and his background?
Yeah, happy to. So Sujal has 20 years of experience as a biopharma executive. Most recently, he was CEO of CymaBay Therapeutics, which seemed to go through every curveball in clinical and commercial development. As he led the company through those phases, there were so many interesting stories and so many hurdles the company and his team overcame. And the drug that they worked on called seladelpar is now approved as Livdelzi. One of the more fascinating turns was as they overcame some of those hurdles, the drug became very interesting to some companies, and Gilead ended up buying the organization for over $4 billion just last year. He has a financial background. He came in as CFO and prior history as a banker, and has just done really well at building good teams. And this is evidenced actually by the drug's first sales quarter under Gilead stewardship exceeded expectations. So he, of course, has a sparkling background. We'll put the full background of Sujal in our show notes, but I think these are the more relevant points to his leadership style that we want to share today. And also, what I think is most important, Shivu, is the healthcare system in this country has been challenged for a while. And as you and I working in the space, and also myself, I can speak to myself as being a patient at times, I find it very frustrating. And there's been certainly some recent events in the space that make you wonder where things are trending. But then you hear about CymaBay and what that company did for its patients. And you think, wow, if there was more recognition for what this company has done and its unwavering commitment to patients, I think there would be better engagement with the healthcare system and more understanding that you can't just paint it with a broad brush and that there are nuances here.
Yeah, I think that's an awesome framing, and this hasn't been a space that I followed closely, and in preparation for this interview, sunk my teeth into it a bit more. This is an incredible biotech turnaround story that you don't really hear that often, and we hope you, as the listeners, really enjoy getting into this one. So, without further ado, we'll get to our interview with Sujal Shah.
All right, Sujal, thank you so much for joining the Drug Dealing podcast today.
I appreciate it. Thanks for having me on, Kenny and Shivu.
So we've already gone through your background, Sujal. I just wanted to set the groundwork for why we're having this conversation. There's certainly been a lot of frustration in this country towards the healthcare system. There's been some recent events in particular, but it's been going on for a long time. As a patient myself, there have been times where even though I work in the industry, I feel like I know how to navigate some things, but it's just so difficult. I met you years ago and I saw what your leadership and your team navigated on behalf of your patients. And ultimately, to see how things were concluded in 2024 with your company being acquired by another in the space. And I just thought, wow, if more people knew how much some fight for their patients, how much better it made me feel seeing how it's such a great North Star for how leadership can be done. Actually, not just in healthcare, in other places too. I felt that it could help others. And so I want to frame that as the beginning for our conversation. And like I said, we've already gone through your introduction. One of the things I noticed when I was reading your background and seeing how much you've already accomplished, there was that small part of me that wondered, are there people maybe in your family who feel, oh, he accomplished all that, but still not a doctor? And I was just wondering if you could talk to me a little bit about your background and how you ended up getting into biotech leadership.
Yeah, for sure. Well, look, in fact, I trained initially as a biomedical engineer with an eye on practicing medicine. So, we chuckle about it, but growing up and the son of Indian immigrants, you're typically given two choices: doctor or engineer. Lawyer, banker, those don't even fall into the mix. It's those two. And so the one I chose, at least initially, was physician. But after getting my undergrad degree in biomedical engineering, I latched on to some research that I found incredibly interesting, did a master's, and spent a couple of years doing academic research, as well as a couple of years at an informatics startup. Some of the core research I was involved in is using biodegradable polymers as scaffolds to regenerate tissue for transplant. So I found it fascinating. later proved to be a fairly challenging area across healthcare broadly. But nevertheless, that was my early path. A few years into bench research, at some point, I felt I couldn't spend the rest of my career in the back of a lab. I love the science, love the research, but felt like I needed to be more on the front lines, more interactive with team members at the highest levels of financing and corporate strategy. So I ended up going to business school as a pivot away from lab research, again, really more towards financial and corporate strategy. Envisioned I would do business development, in fact, in biotech or pharma. Had an opportunity to intern at Roche and later was given a full-time offer to join Roche in BD. But before graduating from my MBA program at Tepper, Carnegie Mellon's Tepper School of Business, also had a chance to meet with a number of folks in the investment banking community. And that became my path away from lab research towards Wall Street as a healthcare coverage banker initially. So I think biotech and healthcare was always in my view. That's the area of interest that I latched onto early on and has been a constant for me. But of course, I've worn various hats from bench researcher to investment banker and eventually as a team member and leader in biotech and drug development.
It's an interesting background, Sujal. And one of the things that I think will come up throughout this conversation is your view on leadership. And I'm curious, there's a lot of stigma around bankers and the way they may act as leaders. And I don't know if there's a long line of folks who believe bankers are necessarily good leaders. But I don't necessarily agree with that. But what I'm trying to understand is what informed your view of leadership? Was it even before your biomedical engineering work? Was it a series of things that happened along the way?
I think many times we all reflect on these types of questions and we're taken back to how we were raised and the family environment we had. There's no question in my mind that having parents, an older brother, a younger sister, folks that just worked incredibly hard their entire lives, it's probably the first example for me of how to lead by example. You do the work and you don't leave early. You don't give other people work that you're not willing to do. I think those are for me at the core of how I think now reflectively about leadership. These are not things I think about necessarily day to day, but as you ask the question and I reflect, I think it has to all start with the fact that, I was surrounded by people that worked hard, a broader family unit that took care of each other and would take their shirt off their back, stand in front of a train for one another at a moment's notice without even thinking about what's that repercussion going to be to my own life if I give up something or if I share something. And those are, I think, some of the core. So coming to the biotech environment initially as a CFO and eventually as CEO, for me, so much of it was about, being part of the team as opposed to some separate persona, if you will, just being part of the team, wanting to contribute, being willing to roll up your sleeves. I think those were some of the things that defined the time I had at CymaBay and perhaps even the time in banking. I joined banking out of business school as an associate. The youngest bankers are analysts, typically out of undergrad. And it's a hierarchical environment. You get more senior, you get to leave a little bit earlier, you do less of the grunt work. Well, as an associate, having never been in that environment, my approach was, I'm going to do all the work that I'm giving my analysts to do. And in fact, I'm not going to leave before my analysts leave. And that was at least my approach to an industry I had no experience in. And similarly, when I went to biotech, I had never been a CFO before. I'd never been a CEO before. And for me, so much of it was to be part of the team and be willing to do everything that you're asking everybody else to do.
One question on that. So we noticed this, I think Kenny and I noticed this a lot, having worked at, bigger companies before. The higher and higher you get up at some of these bigger companies, the further away you get from the work. And, that's not always the case. And that's kind of one of the things, I think both of us like about being at a smaller enterprise, right? It's such a small group of people. It's a more, intimate team environment. Even just the scope of what you can focus on is more narrow. It's limited by the drugs that you're developing. It's limited by the funding that you have. But I guess, how did you set that culture at CymaBay of you as a leader and your leadership team of, not being so far away from the work, right? Like, you get to some of these big companies and there's a bunch of VPs who just don't do any work, right? They're just, they're so far away from the work. They're just making kind of strategic decisions about things. How did you, stay involved, stay close to it? How did you kind of set that standard with your team?
Yeah, Shivu, it's a great question. And inevitably over time, even myself, I had less and less opportunity, if you will, to be in the trenches, to roll up your sleeves. And I think that's just a natural progression of growth. I think it's just a mindset, to be quite honest with you. I never imagined it was such a difficult thing to do. You just have to be willing to do it. And at some point in time, when you have enough resources, enough experience around your team, where you don't have to do, some of that work, you still offer. You still make sure the team understands that you're sincere about the fact that you're willing, to put a slide together. And you have opportunities to set those examples as well, by the way. We had board decks and board materials, and I still made board slides while we were a $3-4 billion market cap company. That didn't change, and you can still do those things. You have to actually care and enjoy it. And so fortunately for me, I enjoyed it. It wasn't something that I didn't enjoy doing. And so it became much more natural. And so it is a mindset. I think it's a mindset of making sure that even as you grow, you're not outsourcing everything, that you find a way not to micromanage. That's an important distinction, by the way, because I think you can lose yourself in doing all of the work, and others don't then feel empowered. But to consciously think about a contribution, and sometimes it's the smallest things. It could be a simple spreadsheet. It could be a slide in the board deck or a slide in the investor deck. But I think it's more making sure that you immerse yourself with your team. And I used to, use the word partnership a lot. And I often use that word consciously to make sure people understand that, while I might have been CFO and eventually CEO of CymaBay, that in the end, I'm a partner to everybody in the organization. And if there's anything that I can assist with and help us advance, that's my job. And that's what I enjoy doing. So, and it does have to be a repeated practice. You can't, that sincerity doesn't exist if you're not that way 90% of the time, and every now and then you tell somebody, oh, hey, I don't mind helping you out with something, right? It really has to be consistently that way for a long period of time.
Sujal, it's so interesting to hear your background on leadership and how you stayed involved. I'm just curious, because CymaBay had so many challenges you navigated. Do you feel as a leader that was your first test, or were there other tests or a series of tests before you became CEO?
Well, I think we had plenty of tests at CymaBay even prior to me being CEO. My first introduction to the company was when they were still private under the name Metabolics. Mm-hmm. And Metabolics had a very rich history of both drug discovery and development, largely in metabolic disease, as the name would suggest, predominantly in type 2 diabetes. Advanced a number of very exciting novel assets, a host of partnerships, including with Janssen, Pfizer, Sanofi, quite a few large partnerships and a very promising early history at Metabolics, but it all hit a wall in the early 2000s when the lead program that was an insulin sensitizer for type 2 diabetes didn't demonstrate a profile in which Janssen believed it had the competitiveness from a commercial perspective. It's not as if the study itself failed, but didn't show the type of profile that would warrant, pouring another 100, 200 million into phase 3 and commercialization. And this was a pivot point for the company. They were faced with, effectively a pipeline that had stalled, mm-hmm. faced with needing to raise additional capital to push the lead program into another indication where there was merit based on the biology, based on the target. but there wasn't an existing investor in sight that was willing to put another penny in the company. And the company had a very rich set of early stage investors, the kinds of biotech VCs that have really supported innovation for many years. Versant, Venrock, Alta, Deerfield, T. Rowe Price, Alliance Bernstein. The list goes on, including J&J's development corporation, their VC arm, if you will. But none of them would put another penny in the company unless we could get an outside lead. This was the company I was introduced to for a CFO opportunity. And to be quite honest, I turned it down. I thought this cannot be my first opportunity out of banking. I needed something a little cleaner, perhaps with a little more of a straight path. And so, in fact, I turned down that CFO role. I had met a couple of other companies where they decided to go with somebody with a little bit more experience. Again, I would have been a first-time CFO at that time. And at some point, weeks later, the folks at Metabolics called back and said, look, if you won't join us full time, we know right now you're just consulting and contracting. Would you be willing to contract as a CFO and help us raise this $30 million we need to keep the lights on, to keep the company from bankruptcy, to fund one additional study and a new indication for the lead program? And I said, sure, I certainly have some time on my hands and I'd be happy to do that. Didn't realize a year and a half of my life later, searching the ends of the earth for capital. So you talk, Kenny, about being tested. So not just myself, the remainder of the team at that company were clearly tested with having to find some way to raise capital. And it was not easy. And after a year and a half searching the globe for a new institutional lead in order to get existing investors to participate and support this financing, we struck out. We couldn't find a new institutional lead. And the company was about 3 months away from bankruptcy when, I sat, I think it was in my car at some, parking lot, just thinking about all those folks that were working for the last year and a half inside the company just to try to raise this capital. They all could have left. They all could have left and found another job in biotech. In the Bay Area, we of course have a rich industry in biotech, and each and every one of them, frankly, could have just bailed. And of course, some of them did because they have to make sure and pay the bills and take care of their family. And I don't consider that a negative whatsoever. But for those that stayed, I sat in my car thinking, how do we give up? How do I give up? Just because I'm a contractor, these folks had just put their heart and soul into it, and I couldn't, conceive of just walking away. And so I had an idea for a financing avenue that I had seen once before when I was a banker at Citigroup. An analyst there had left for a company in the GI space and had actually raised $20 million of capital as a private company from retail investors, high net worth individuals where the average investment is $50,000 to $100,000 per person. Now, remember, we needed $30 million to fund one more study and get to that readout. So $20 million from retail is not an insignificant number. In fact, it doesn't happen very often. And even at that time, I could only think of a couple of examples. But we had to try. And so I made a call to this firm called National Securities that has an independent network of brokers that feed into their systems and the national security umbrella. And we went out and pitched to these brokers the story of a study for this lead program in chronic gout, a profile that we thought was fairly exciting based on the biology again. And sure enough, that helped us entice some of the existings to put in the equivalent of about 10 million of that 30 million. The retail investors put in another 17. I put in some venture debt into that. And there are stories and stories about how there were dozens of times all of this was ready to fail, that it all would have fallen apart. A singular detail, either from the retail piece or the institutional piece or the venture debt piece. We also had to convince JJDC, the VC arm of J&J, to forgive a $16 million in a convertible note the company gave them and take $3 million of equity credit in the pro forma company, which was going to be at an incredibly depressed valuation from the prior rounds. And so every day I worked with this team as a contracting CFO. In fact, something would come up that almost killed the entire deal, over this 2-3 month period. And I joked with the CEO at the time and just said, maybe we shouldn't come into the office for a couple of days just to make sure this gets done, because every time we come in, we're putting out another fire. But it was, at the end of the day, just a relentless effort by an entire team to not give up. And we raised $30 million, in fact, $32 million, when the company had 3 weeks of cash left on the balance sheet. We were selling lab equipment to make payroll for the people that were still in the company. We're selling furniture in the office. It wasn't even really nice furniture, by the way. It's just anything we could get money for to pay, to take care of payroll and to take care of the people. And the company was less than 15 people by then. But we pulled it off in a rather, I think, miraculous financing just to keep the lights on. And of course, 12 years later, we finally got a drug approved. But, those were early days tests, well before I even joined as a full-time CFO and before I became CEO of CymaBay. And it wasn't just myself tested. Obviously, it was the entirety of the team. And so there was an early grit and resilience this team showed that later, again, proved to be necessary and valuable.
It's so fascinating, Sujal. Born of fire. I hear this story and I feel like the challenges, I think of the specific clinical challenges that help you get your drug approved, but I didn't recognize how many challenges you had, not even as a full-time employee. You were dealing with these things. A lot of contractors have very different dynamics with their roles. That's so fascinating to hear, and explains a lot about your view of camaraderie and how you get through things, and that you had a team in place as well. And selling furniture to make payroll, it's not as common in biotech, and so it's such a fascinating nuance to hear. Maybe, Sujal, what would be helpful now is, since you've kind of laid the groundwork for the beginnings of your work with the company, can you tell us a little bit about this drug we've mentioned already, seladelpar? I believe it had a serendipitous beginning in PBC. You mentioned some of that as well, and of course, this is the main reason why another company acquired the organization. Can you walk us through a little bit about the early curveballs of seladelpar and how it started in PBC, please?
Yeah, sure. So, seladelpar is a selective and potent PPAR delta agonist. And PPAR delta is a nuclear receptor target that's involved in a variety of different processes. And agonizing PPAR delta upregulates and downregulates a host of genes, for example, involved in lipid transport, metabolism, involved in inflammation and fibrosis. Some of what was known about delta and what excited folks in the drug development industry about PPAR delta is that it also promotes fatty acid oxidation. Mm-hmm. And so PPAR delta initially as a target in the field was one that people thought could be utilized and developed for lowering LDL cholesterol, lowering triglycerides. And so in fact, seladelpar was an asset that CymaBay received from Janssen. In the early days in which they were developing another asset, a very promising insulin sensitizer for type 2 diabetes, the company outlicensed the rights to that asset in a fairly competitive process. And of course, in the end, Janssen was the one who won that process. But predecessors of mine at the company had the foresight to not just ask for upfront and milestones, but actually ask for a clinical quit in return for out-licensing that insulin sensitizer. That's actually where seladelpar came from. So Janssen had simply completed phase 1 at the time, and the asset was phase 2 ready. CymaBay, again, under the name Metabolics as a private company, ran a phase 2 study in mixed dyslipidemia. Mm-hmm. And this was a study run in monotherapy as well as on top of atorvastatin, or Lipitor, the Pfizer drug for lowering LDL cholesterol. And it showed the profile you would imagine with this mechanism: LDL lowering 20 to 30%, triglycerides 30 to 40%. But something had changed in the development path in mixed dyslipidemia. There were a number of things that changed. One, there were more competitive assets and mechanisms out there for lowering LDL. You had the advent of PCSK9 inhibitors. For triglyceride lowering, you had the advent of novel fish oil compounds. And then the regulatory environment also changed. The FDA wanted to see pre-approval outcome studies for novel targets in garden variety dyslipidemia. And that meant for a small company, a large outlay of capital just to get, a drug through development and to potentially approval. So, the company effectively shelved seladelpar for a period of time. as I mentioned, the recapitalization of the company that I helped lead before joining full time as CFO was to advance another asset in chronic gout. Seladelpar sat on the shelf for the first couple of years. It had always been a compound we wanted to put back into development. In fact, always something that we thought would be promising. The question was what indication? And so in the early days, two indications popped out. One was related to lipid disorder. It was homozygous familial hypercholesterolemia, or HoFH, which was a population of patients where statins, really didn't have as much efficacy, and other compounds also struggled. And one other area was primary biliary cholangitis. And again, I have to give credit for our then CMO at the time. His name is Paul Boudez, who, had the foresight to observe and study the phase 2 data in dyslipidemia and observe that in those patients, while they didn't have a cholestatic liver disease such as PBC, they saw a decrease effectively from the high end of normal to the low end of normal in a liver biomarker called alkaline phosphatase, which is a marker of cholestasis or impaired bile flow. And this observation was one that led him to believe this could be something worth exploring in PBC. At the time, there was a company, Intercept Pharmaceuticals, developing an FXR agonist in PBC first, eventually also in Nash, but initially in PBC, where the primary marker for efficacy was lowering ALK phos, which is elevated in patients with PBC. And so this is some of the early origins of what led us to advance seladelpar into PBC. Again, this is all in the time at which I was still CFO. So the prior CEO, Hal Van Wert, the prior CMO, Paul Boudez, many others on the team, Chuck McWhirter, our chief scientific officer and head of development throughout all of my 12 years. I think credit to this team for thinking about the advancement of this asset in this area that later proved to be quite promising.
So it sounds like, if I'm understanding it correctly, there was a realization that, hey, this financial hurdle we have to get through to, run a outcome study even before it's a pivotal study in mixed lipidemia was just not going to be a viable path for us. So what are, still high unmet need areas that are lower cost, potentially faster time to market avenues for this drug? Is that right?
That's exactly right. One of the things that I always felt, particularly leaving banking to industry, is in a small biotech company, you have to find a way to control your own destiny. And developing drugs for large primary markets are challenging, just given the capital outlay and then even the commercial footprint necessary if you're successful. I always felt like you needed at least one asset in at least one indication that you could take all the way to patients. And so, Shivu, you're absolutely right. Whether it was HoFH and eventually, in our case, PBC, that was an indication in which we fully felt we could fund development, fund applications for approval, and frankly, even fund commercialization. Another principle I have is there's no real bluffing in biotech, whether it's raising capital or executing a business plan. We're dealing with an industry where we are fortunate to have the opportunity to do work to advance people's lives or improve people's lives. I don't believe we're in an industry where you bluff. And so you have to know that you've got a strategy and a plan to bring the drug all the way to patients. And I think that's something really important for small companies. Absolutely, licensing and partnerships are critically important. But to be a small, viable company, we always felt like you needed at least one asset you could take all the way to patients. And so for all the reasons you outlined, Shivu, PBC was one of those exciting areas for us.
And maybe just for the audience, could you tee up just the level of capital you would have needed for the mixed lipidemia path versus kind of what you ended up going with, just so they can get a sense of, these outcome studies are not cheap. They're long, large studies. They're just massive. And I think Kenny and I have talked about this in kind of previous episodes a bit. We've touched on it a bit, but that's one of the big challenges to developing drugs, for the massive killers in our space, right? Like heart disease, ASCVD, all that type of stuff, right? Is these are really big, long studies that, take a ton of time to get drugs approved. And there's only a few types of companies in our space that are well equipped, both operationally and financially, to kind of do these things. And so you all had to, make a strategic decision.
Yeah, absolutely. You make some great points. And I think over perhaps the last 5 to 10 years, there's been more light shown on the fact that we need to find ways to support development for some of these large chronic diseases where effectively development fell off a cliff because of these dynamics. At least 12 years ago, that really wasn't still the conversation. So you're absolutely right. It was really hard for us to stay in these areas. It would have been several $100 million. And remember, at that time, we were a sub $100 million market cap company. This recap that I discussed briefly was at a $20 million pre-money valuation with 32 million raised. We were a $52 million post-money company. So we went public and started trading eventually on NASDAQ post that private financing at a sub $100 million market cap. There was no way we could raise 3x our market cap in capital to fund these studies. So in order to do a proof of concept in PBC, we're talking about 25 million to do a proper phase 2 dose ranging proof of concept study. So, magnitudes less than what we would have in an area like mixed dyslipidemia.
So Sujal, thanks for walking us through the beginnings of that. Help me understand about what time, what year are we in right now as we look into PBC, and were there any other thoughts for any other liver diseases, things like Nash, MASH? When did that start to enter the horizon? At some point, I think there was another disease you were evaluating too called PSC, primary sclerosing cholangitis. Can you walk me through the next steps?
Yeah, so it's about 2015 that we believed it merited moving seladelpar into phase 2 study, again dose ranging in PBC, where we were looking predominantly at the biomarker of alkaline phosphatase, but of course measuring a host of things, including some of the symptom burden that patients experience, particularly in PBC, where there's a tremendous amount of itch or pruritus experienced. But it's about that time in 2015 that we moved into PBC itself. That was about a year before Intercept was able to get their FXR agonist approved in PBC, based on a surrogate endpoint that included alkaline phosphatase and bilirubin. And because there was now an accepted, validated endpoint, particularly a surrogate endpoint, as opposed to outcome, to get Subpart H accelerated approval, we were first and foremost focused on PBC. Again, the biology, we then, from the observations in the early days in the dyslipidemia study, the teams did the work to understand the mechanism of PPAR delta as it relates to alkaline phosphatase and bile acids, and learned that we in fact inhibit synthesis of bile acids. Inhibit the expression for the enzyme that converts cholesterol to bile acids. So you lower bile acids. That's how we're lowering alkaline phosphatase and the bile acid burden in the livers of PBC patients. So the biology, the company elucidated and felt strongly this was a path forward, and this should be our lead path forward. And through some very successful phase 2 development programs and studies, felt like we eventually found the right dose that balanced both meaningful efficacy and safety. We had even hiccups along that path, by the way. There were higher doses that had been very safe and well tolerated in mixed dyslipidemia that unfortunately in PBC patients, perhaps something to be said about the livers of these PBC patients caused, some transaminase elevations, transient, asymptomatic, but enough to tell us, we need to pause development in the early days of phase 2 development in PBC, 2015, 2016, and go back into the clinic at lower doses. And when we did that, we didn't see those elevations. In fact, because of the anti-inflammatory mechanism, we saw decreases in transaminases for many patients that had elevated transaminases. And so we saw not only a positive anti-cholestatic effect, we saw a very nice anti-inflammatory effect, and found doses that we certainly believed would have the meaningful efficacy as well as a balanced safety profile. And so there were those challenges early on, but these are around the 2015-16 timeframe. You asked about other indications. And so once we felt we clearly had an opportunity to move from phase 2 into phase 3, had an end of phase 2 meeting, outlined a development program that really mirrored what Intercept had done to get obeticholic acid, their FXR agonist, approved in PBC. We then continue to think through the profile of seladelpar, now being more focused in chronic inflammatory liver disease. And to your point, Kenny, there were two others that were quite interesting for us to at least develop some proof of concept data around. One was Nash, now commonly referred to as MASH, or metabolic associated steatohepatitis. Some of the reasons for this is we know that PPAR delta is in fact expressed in all four major cell types of the liver, not just hepatocytes, but cholangiocytes, which line the epithelial cell lining of the biliary tree, as well as Kupffer cells, the predominant inflammatory cell type in the liver, and then stellate cells, responsible for laying down extracellular matrix, collagen, the fibrotic cascade. And given what we knew about the biology, we felt it was very interesting to study seladelpar in NASH-MASH, and had actually initiated a phase 2 paired liver biopsy study in NASH-MASH, where the primary endpoint, in fact, was a non-invasive endpoint. It was looking at liver fat reduction at 12 weeks versus placebo. And we had several doses of seladelpar. But the study itself was a 52-week study. So even beyond the primary endpoint that was 12 weeks, the idea was to read out the end of treatment biopsy at 52 weeks and compare that to the screening biopsies, so that we could not only get a measure of this 12-week liver fat reduction, but also look at the NAFLD activity score and the effects of seladelpar on fibrosis in what were largely F2 and F3 NASH patients. And so that was a study that we actually executed while we were moving seladelpar through phase 3 recruitment and development in PBC. And at some point, even as the NASH phase 2 study was coming to its conclusion, we had also thought of another cholestatic liver disease like PBC called primary sclerosing cholangitis. It is a different disorder, and there are some key hallmarks that differentiate it: more aggressive, much more fibrosis than PBC. But that was another area of high unmet need, where in fact patients had no approved options. PBC at the time we were into phase 3, there were only 2 approved drugs. PSC had no approved drugs. So again, looking, and neither did NASH at the time. And so we're looking around at that landscape of chronic inflammatory liver disease, areas where the biology and what we knew about the clinical profile made sense to develop. That was always important. And that's important for all of us in the industry.
Really interesting views on the clinical development of seladelpar. I wanted to go back to one piece before we get into the next steps of your clinical development. When you said you had to go back to PBC patients, realizing their livers function fundamentally differently, so you have to dose a little lower, were there concerns about seladelpar's therapeutic index and potential efficacy? Or did you feel, based off your modeling, lower doses would be okay? I'm just trying to understand, again, as a team and navigating this on behalf of patients, were you concerned about dosing too low at the trade-off of efficacy?
Oh, for sure. Look, I think early signals from early clinical data only tell you so much. Certainly, we had some confidence we would see meaningful efficacy, because what we were seeing at those higher doses that led to a few of these cases of transient transaminase elevations was extraordinary efficacy. In fact, many of the physicians involved in our study, thought leaders in PBC, really encouraged us as we halted the first ever phase 2 PBC study because of these transaminase elevations in a few patients. They really encouraged us to go back in the clinic at lower doses because of the extraordinary efficacy we were seeing at higher doses. So, but there's always uncertainty. The first study that I mentioned to you, where we saw these few transaminase elevations, we stopped that study ourselves. Of course, we had a DSMB, and so we were developing the drug responsibly, but we stopped that study because we knew we couldn't put patients at risk. Going in at lower doses, I think to your point, Kenny, yes, we had at least evidence in other patient populations, evidence from phase 1, that those lower doses would be safe and well tolerated. But you just don't know until you actually do that work. So one of the things we did, Kenny, to your point, is the next phase 2 study at lower doses. We did that study as an open label study. And, while we typically want to do, blinded studies in drug development to maintain objectivity, we already had, a partial blinded study that we had done, the one we stopped early. So we thought we had some, meaningful observations. We did the second study at lower doses as an open label study. So we could, more specifically day to day evaluate both efficacy and safety. And if we needed to, if we continue to see a safety signal, pull the plug on the development program entirely. Luckily, we didn't see that. We saw very good safety and very good efficacy even at those lower doses. So we took what was an open label 12-week study and extended it to 52 weeks. And so eventually we actually dosed even longer and developed longer safety and efficacy data at the time.
Sujal, I think it's so easy to gloss over this, but there are nuances there, right? Knowing that you have to go back and reevaluate a couple doses through this nuanced approach with an open label, did that impact your OpEx? Did you have to go back and say, okay, here's our cash runway. We have to go back and look at a few things. And then how did you communicate that to investors? Because there's always a skepticism in the community externally.
Oh, for sure. Look, all this time we're a small microcap public biotech company. And we were living the life of financing the company, frankly, year to year, study to study. And so when you have an announcement, as we had, that we were halting a phase 2 study in PBC, you might imagine what happens to the share price and what eventually happens to access to capital. Mm-hmm. So once again, you talk a little bit, you asked me early on, were we tested before what later proved to be a pretty significant test in our phase 2 Nash study as a company? Oh, absolutely. We were tested. We were tested every year. How do we creatively raise capital to fund that next study? How do we find the investors, willing to listen and willing to take, the risk for the next development, given the potential promise of the asset? So over and over again, we had to think about OpEx, how to creatively raise capital. And I think the underlying principle, these are things I reflect on only because we're having this conversation. They're not the things I reflected on while we were in the trenches. But I do think about it. And of course, I hear a lot from our investors that were with us through thick and thin, even in the worst of times. And I think so much of it had to do with being transparent, being honest and transparent, and sharing as much information as we possibly could share publicly. That's also a balance. But I always felt we had to err on sharing as much as possible. That may have meant it was going to be harder for us to raise capital, but so be it. I think you gain so much trust from obviously all of our stakeholders, patients, partners that are dosing their own patients in our clinical studies, service providers, of course, investors, by being fully transparent. So those were some of the principles early on. It made it harder to raise capital. Sometimes we had to be grittier, but we found a way to, share the opportunity relentlessly with investors. And this was also a principle of mine. As I transitioned from CFO to CEO, given my early days were as a CFO in the company, even as CEO, I talk to investors every day. You might think a drug development leader is not talking to investors every day. Hopefully they don't have to, but sometimes you do. And when you're living the life of a microcap company, you have to, because you're going to need more cash. You're going to need more capital, particularly if you're successful, but even when you hit bumps in the road. And so I think these were some of the things that we did well in those early years, is just manage OpEx, but also manage to bring in new investors, additional capital, just to get us to each next step.
Excellent. And one last question on this topic, because I am curious myself, the open label with regulators. One thing we haven't talked about yet is the FDA. What were your interactions like? Did you feel that, this is not a big indication? Did you feel the FDA was supportive of your development plan, that if you go lower dosing before you head into registrational, that's the right way to go? Help us understand what that dynamic was like, because the external community will ask of that as well.
Yeah, sure. I think for regulators, of course, that your registration study will have to be blinded. That cannot be open label. And so the phase 2 is really up to the innovator, up to the company. The regulators wouldn't tell you have to necessarily do this phase 2 as a blinded study or open label. You just happen to then carry the risk yourself into phase 3, perhaps if you do it open label and you don't really know. And that's something that perhaps was harder to explain even to investors. The big nuance here, the reason why it was a relatively easy decision for us, is the endpoint was an objective endpoint. It's a blood-based liver biomarker. And so even in an open label study, you're not so concerned about the fact that you may be tainting your data. There's something subjective between placebo and treatment. It's a blood-based biomarker, and that made it a relatively easier decision for us and an easier story to tell investors as we then had success in phase 2 and then moved into phase 3.
Fascinating. So, Sujal, I think a couple of years has passed now. We've moved a couple of years beyond 2016 or so. We're heading into Nash. Perhaps the next clinical curveball, we've already hit a few. Perhaps the next clinical curveball we can talk about is what happened with the Nash study. So I'll frame a little bit up here. I believe it was, summer 2019, right around June or so. I think your phase 2 study failed to meet the primary endpoint for MRI-PDFF, the liver fat reduction.
Right.
And I think it's also important to highlight, because you were a publicly traded company, what the perception was. I think perception is interesting because you have to deal with that as well. Patient, investor, so many external stakeholders. I believe your stock was more than halved at that point. It went from $13 to $6. And I'm trying to understand what it's like to be a leader and go through something like that. So you still had PBC ongoing, but you had Nash. And I think what's also important for people to remember is as some of the companies, AbbVie, Gilead, started to make significant progress in curing HCV, there was a lot of excitement for what is next in liver.
Right.
And Nash MASH became one of those things where I believe it's still to this day, in the next few years, going to be the likely leading cause of liver transplant. This is how significant a problem it is. And so there was a lot of excitement in this time period for anyone who has a Nash study. I remember if you made an announcement, you got a stock bump. It's like AI today.
Right.
Oh, I have an AI ML machine. You're going to get a bump. And there were so many companies who followed that fast wave. So help me understand, when you had excitement around the Nash study, you were communicating, you had so many positive potential guides that this could work, what it meant to go through this curveball, how did you navigate it, and what did it mean for PBC? Like, did you feel there were ripple effects of this?
Yeah, great questions. Look, I think through that time, we tried as hard as we could inside of CymaBay, and I applaud the team for this, to maintain their North Star in the lead indication where we had meaningful data, and we were never going to abandon the thesis around PBC. The exploration of seladelpar and Nash, again, had to do with following the science, understanding the biology. PPAR delta is anti-fibrotic. We saw this in animal models. We had run, not just preclinical animal models in various settings, but particularly in Nash models. And so some of what was observed really led us into this phase 2 study. We tried really hard not to get swallowed up into the hype. But to your point, Kenny, when the outside world sees the profile of your compound, its mechanism, the biology, and gets as excited as you are, of course, then you tend to have some of this built into your evaluation. And so we had a thesis largely around the anti-fibrotic effects of PPAR delta, but also around the potential lipid effects and potentially liver fat reduction, right? Just given the fact that it promotes fatty acid oxidation, not just in the liver, but in other tissue where PPAR delta is expressed, including adipose or fat. So, we were true to the biology and the science, moved in to Nash with excitement. And yeah, we were quite disappointed when at 12 weeks, we didn't see the liver fat reduction that we had hypothesized. We obviously created that study with that as the primary endpoint, a non-invasive endpoint, that was early on to give you a signal. Weathering that storm, I would tell you, was only something that we managed through because we knew we still had a drug. We had a drug for patients with PBC. And as drug developers, there's no question there's excitement when you can move forward into indications that obviously affect many people. But I'll be very honest with you, Kenny, I don't know that there was anyone inside of CymaBay who felt like Nash patients themselves were more important than the PBC patients. And so as a drug developer, and this is, you talked a little bit at the outset around, folks from the outside that may have a tainted view about the pharma industry and the biotech industry. I think it's the case that those folks doing the work day to day, they're not so focused on the dollar signs of the therapeutic opportunity or how large the therapy. It's all about, can we help 1, 12, 100, 1,000 patients? And, the satisfaction I can tell you now, years later, having gotten seladelpar approved for PBC and never approved for Nash, I don't think is any different for me personally. Knowing that the PBC patient population is smaller than Nash doesn't make a difference to me at all. So I think in those moments where Wall Street, sells your stock because now the bigger opportunity is not available. Yeah, of course, there's a reason for that. The valuation of the company is tied, to the size of these opportunities. But in terms of what we inside the company care about, it's developing a drug to help patients. Doesn't matter how many there are, frankly. And so that, I think, is the core in which, dealing with the pressures of the stock and unhappy investors. Those are things hopefully I had the opportunity to do to shield everybody else in the company day to day. I felt like that was certainly my responsibility, while everybody else remained focused on development in PBC and even getting to the end of the Nash study. Now, specifically in the Nash study, as you mentioned, even after the 12-week MRI PDFF data that failed to meet its primary endpoint, we were still encouraged by what we might see in liver biopsy. There was, as you said, a lot of excitement in Nash, and at the time, a lot of hypothesis that when you have drugs that lower transaminases like ALT, AST, that those could be tied to reductions in fibrosis. That there was at least some data, and Rohit Lumba at UCSD actually had published a paper that we talked about with investors all the time, where it observed this correlation between ALT decreases and decreases in fibrosis. So we maintained, at least within the company, some excitement that we might see positive results on liver biopsy, which eventually we in fact did. Although I think we're going to come to some of the curveballs here that almost brought the company to its knees.
And I'm curious, Shiv, Sujal, just on this part here with this, did you have to make any organizational changes? Because Nash being such a big disease, you might have had a bigger footprint in place. Was there anything you had to change just organizationally, or you were able to just continue as is?
Yeah, at the 12-week primary endpoint readout, we didn't have to make any changes. We were already a very lean company. In fact, I want to say we were still maybe 50 full-time employees, 60 full-time employees, as we were, at that time, continuing to enroll in PBC. Mm-hmm. So at the 12-week readout, we really didn't have to make any organizational changes. We were a very lean organization.
Okay. Excellent. So this is summer of 2019, June 2019. We're heading about 5.5 months forward now, right before a major holiday. It's November 25, 2019. And this is the next clinical curveball I wanted to get into. And this is on PBC, the indication we were talking about. Monday of Thanksgiving week, clinical development is terminated for Nash and PSC, which was recently initiated, I believe, right, Sujal?
Right.
And PBC was put on hold. Reasoning, atypical histologic findings. I want to highlight what this meant externally. We're talking here in a vacuum. What did this mean? Well, your stock was around $6 or so. The market took it to $1 to $2, if I remember correctly. Mm-hmm. So what this means is, even if you don't understand the diseases as well, the investor community basically was writing off CymaBay. That's what this news development meant. And so we're going to get into this in greater detail. So can you help us understand, you had this Nash, it didn't work out as well. Okay, things are moving forward, fine. But then you had this PBC development right before a major holiday. Seemed unexpected, Sujal. Can you walk us through how things developed and what you learned at that day?
Yeah, Kenny, one very important clarification. The atypical histological findings were actually the 52-week biopsy review from the Nash study. So just to paint the picture for you, in November of 2019, we had just been fully enrolled in our phase 3 registration study for PBC. That blinded study was ongoing, really no hiccups or challenges from that perspective. And as that study completed enrollment and was effectively 1 year away from reading out top-line 52-week data in PBC, that if positive would have led to filing for approval and eventual approval in PBC. We were also at that time nearing completion of the 52-week dosing period in the Nash study. So as you mentioned, the 12-week endpoint on liver fat reduction didn't meet its primary, but those patients stayed blinded and continued to receive seladelpar all the way out to 52 weeks. And it was the 52-week biopsies in Nash that were being read that highlighted this atypical finding. Okay. So quite literally over the course of a weekend, we are alerted to this pathology by our study pathologist involved in the Nash study. Okay. And they were incredibly alarmed. I can't tell you this was a casual update of we're seeing something unusual in these end of study biopsies. It was a fairly significant alarm. And these were, one of the reasons I think I told you offline before, I didn't want you to introduce me as a distinguished biotech executive, because I don't know what the word distinguished actually means. I say that sincerely. I'm not just joking. But we had otherwise, quote unquote, world-renowned expert liver pathologists involved in our study, folks that are well published, part of the Nash CRN, clinical research the NIH CRN, part of the clinical research network at the NIH, whose scales everyone in NASH/MASH uses even today to evaluate liver biopsies. These are the folks we had involved studying these liver biopsies. And when they read the 52-week liver biopsies, they were blinded to 3 different treatment arms versus placebo, but they knew these were end of treatment biopsies. We had not scrambled the screening biopsies in the mix. This was not something anyone did at the time, really. So they knew these were end of treatment biopsies, and they looked at the first set and felt like in 30% of the first, dozen, and then the first 3 dozen biopsies they read in the matter of 2-3 days, 30% had pathology they believed atypical of NASH. So what does that mean, atypical of NASH? So, they're describing pathology that is not believed to be part of the pathophysiology or the development or etiology of NASH. And in our case, this pathology, without going too far into the depths of the detail, was largely being referred to as it was interface hepatitis or periportal inflammation. There was also some pathology they felt was atypical, like bile duct injury, presence of plasma cells and eosinophils, even some, they believed, vascular changes that all could mean that either every one of these 30% of NASH patients had some other disease like autoimmune hepatitis or PBC or PSC, which would have been completely unexpected because you would screen those patients out. There's no way these world-renowned pathologists wouldn't have screened out 30% of patients that all had NASH plus some other disease. Yeah. So the only remaining hypothesis was that seladelpar was causing drug-induced liver injury and potentially, drug-induced autoimmune hepatitis. We had no way in those moments to prove that the pathologists were right. We also had no way to prove they were wrong. And despite the fact that these very patients, these 30% flagged as having this atypical histology and potentially histology suggestive of drug-induced liver injury, despite the fact that they didn't have spikes in liver enzymes. Typically when you have liver injury, you see transaminases spike, early sign. ALT,
some of the
AST.
correlates we've talked about, you get an indication this is where it's trending. Yes.
We didn't have those. There weren't clinical symptoms or clinical events. We didn't have patient deaths. We didn't have patients complaining of flank pain, which is obviously often considered to be liver injury. We didn't have those signals. However, even in the absence of those signals, pathology trumps all. What the pathologist sees in your tissue effectively trumps all. Okay, this is what is typically largely accepted in the field. And so we couldn't prove they were right. We couldn't prove they were wrong. We had only one recourse over a course of a weekend, which was to shut down all development. We, of course, shut down the Nash study, unblinded it. And rather unfortunately, in those early moments, in those early couple of days over the weekend, while the number of these quote unquote unusual findings was pretty similar between the 2 lower doses of seladelpar and placebo, they were actually 2x the amount in the highest dose of seladelpar versus placebo. So even that had the potential suggestion that it was related to seladelpar.
Yeah.
Right. So this is the unblinded data we saw. Years later, people tell us, well, why did you stop the studies? It's unusual. If you didn't have transaminase elevations, you never stopped the studies. Live a day in our lives at that moment in time.
Yeah.
And tell me you wouldn't have stopped the study.
And worry about patients.
Exactly. Drug development yes, we're trying to improve patient health, improve their lives, but do no harm.
Mm-hmm.
And, people ask me all the time, what was that weekend like? It was without question. What was that decision like to stop the Nash study, the PSC study, but also halt the PBC study, the phase 3 PBC study that was fully enrolled a year from data that had no safety signals of concern, but it's the same drug we're studying. We couldn't prove that it wasn't doing the same thing in these patients, some sort of potential harm. And if you can't prove that, then you only have one moral obligation, which is to stop it all. And I knew that that would mean not just a potential end to the seladelpar program in any indication, including PBC, but because we were largely a one-product company, it was likely an end to CymaBay. And that was, of course, a gut-wrenching decision, one that we followed with laying off 2/3 of our company before Christmas in 2019. The other side of that coin is we were never going to live with having asked patients to just trust us or stay on the drug until we figure it out. I was never going to live with the idea that we put somebody's, mother, sister, daughter. PBC is largely a disease affecting women. 9 out of 10 patients are women. We're not going to put these patients' lives at risk until we better understood this. And if we never understood it again, it was going to be game over.
I am curious on the personal toll because of what you expressed as your primary goal of taking care of patients and making a difference for that population. I am just curious, taking a quick detour, that holiday for you personally, you had Thanksgiving that week. Are you able to turn it off? I am asking candidly as someone who aspires to be a leader. I don't understand how to separate these two. How do you handle and balance both, knowing that you have a family?
I'd say it was impossible, really, to be honest with you. I think you just wake up every day and try to do your best and try to remind yourself of all the various important things in your life. But it wasn't just me. I would tell you, all of us in the organization, and of course, I'll talk about my situation and my feelings, but I think we all had these types of feelings. I personally went through a period of time in which it felt a lot like I lost a family member, maybe even lost a child. And of course that would be worse, but that's what it felt like. I would wake up in the middle of some nights thinking it didn't happen. So these are the things our human brains can do to shield us from pain. I quite literally woke up some nights thinking it didn't happen. I spent many days paranoid that somebody had done this on purpose, that this wasn't just an accident or an interpretation or false interpretation. I went through days where I felt like somebody conspired against us.
Wow.
And again, that's just my mind, unfortunately trying to perhaps shield me from pain, but it didn't feel like it. Those were tough days, no question about it. We pour our lives into these efforts, and yes, absolutely, it's to make an impact in the world, and that doesn't mean that it's void of some selfish motivation of trying to be successful and leave a legacy. Those things are all part of it. But I will tell you that in those moments, when I once thought that this journey was about succeeding, making investors money and our employees and myself money, all of these various things, notoriety, when it was all gone in a heartbeat, my realization is what hurt the most was a loss of purpose. And losing that loss, having a loss of purpose where, every day you went into work for the last 12 years, in those most difficult moments, some of which we talked about, nobody wants to give the company a penny to raise capital, stock price going up and down, fighting, tooth and nail just to raise capital. In the moment it's all gone, you realize what hurts the most is your purpose, your reason for waking up every day and doing that work. And it was nearly impossible for me to separate that from my personal life. I had a hard time finding joy, in my young kids' activities and their games. And of course I did it. And I did my best to be present, but I would tell you it was nearly impossible for me to separate those emotions.
It's interesting you mentioned purpose. So it seems like with this development, I'm sure there were organizational challenges And it seems like you would have to rebuild your purpose because you have to find the answer to this, right, Sujal? I don't want to lead. Help me understand. What do you do when you go through this news? You communicate. Dust has to settle for a major holiday. What do you do now? What do you and your team try to figure out to see what to make sense out of this?
Yeah. So, like every other day, it's doing a number of things in parallel all at the same time. We knew immediately we had to reduce the workforce. That was, again, one of the more gut wrenching experiences I ever had in my 12 years at CymaBay. We were about 65 employees. We laid off 45 of those employees because we didn't know how long this might take for us to figure it out, if ever. So that was a process we went through. In parallel to trying to find other experts that might help us down a path of trying to investigate these findings. Within even the first few days, however, there was an arbitrage hedge fund in New York City, not a biotech investor, but simply an arbitrage hedge fund that saw that this was a company now trading at $100 million market cap with $200 million of cash. So effectively, we were trading at $1 a share with $2 of share of cash. Proceeded to buy 10% of the company at $1 a share and now became the largest shareholder of the company with an objective of forcing the company to liquidation. Because then for every dollar he spent on a share of CymaBay, after liquidation expenses, maybe he gets another $75 return. So 75 cents return, right? So that was this person's singular objective. And it attracted a host of others like him who felt like they could just ride his coattails. So we're laying off 2/3 of the company. We're desperately trying to find experts that might be willing to help us investigate these findings. Dealing now with a number of new investors who just want to see liquidation so they can make a quick return on their investment. All of a sudden, we're dealing with a global pandemic because now by the early part of 2020, we're all at home. And I don't know how to describe it other than you wake up every day trying to put out another fire and as many as you can to just keep moving the company forward. And in those early days, I had plenty of people, even a board member of mine say, Sujal, you're young. This is over. You need to just get out of here and go find something else to do. Don't sit around here. You need to just leave.
I'm curious, were you ever close to taking that, to saying, I think I'm just going to end it here and find the next thing?
No, not at all. It never thought in my mind. And I will tell you something that I think is a principle that not just myself, but a number of the other team members showed, not just then, but even as I mentioned to you early on in that first recapitalization back in 2012, 2014 to keep the lights on. My principle is, you talk about leadership. My thought is, you're either the last person to leave. You have to be the last person to leave a burning building. You have to be the one there all the way to the end. So it never, despite people telling me, despite investors, I had investors who had lost tens of millions say, Sujal, you and your team did an honorable job. You communicated so transparently as soon as you saw these findings. We rarely see, teams so transparent in a situation where they even don't know all the facts. I had big institutional investors tell me, whenever the dust settles, please give us a call. We have other companies for you to go run. And even with all of that, I never once thought about just leaving that building until being effectively waiting until I was the last person out.
Yeah, go down with the ship. This oh, captain, my captain.
Sujal, we have some similarities. I also have an MBA, and that's not what you think a lot about resume bullets. And it didn't seem like that didn't even register in your mind. So you didn't worry as much about, if this became a shell company and you had to liquidate or anything like that. You were just going to finish it through no matter what. Help me understand now the plan forward. You have this signal and you have to do a review of all the data, correct? You have to understand where did this come from? And maybe we can talk a little bit about the issues when you have humans reviewing tissue samples versus some of those objective markers.
Absolutely. So, among the various initiatives, including considering merging into another company and giving the cash to, some other company with other assets, in parallel to all of that, we felt like we had to look for some answer. We had PBC patients who were in tears that we had to stop the PBC study, that quite literally told their physicians they didn't want to give back the drug, that they were willing to take the safety risk, if that was the case and whatever happened in our Nash trial. Many of them, again, in tears because they'd never felt better than when they were on seladelpar. And you talk about motivation. So I'm sorry to go back a step here, but
no, please.
Had a patient in her family reach out to me. We had several hundred patients in 20 countries around the world, and it just so happened one that reached out to me lived 10 minutes away. So I met them, and to this date, I'm friends with the entire family, including the woman who was in our study at the time. But they were devastated because they said, this drug didn't just change her life; it changed her husband's life, her 3 kids' lives. They felt like, mom may never need to have a liver transplant. How do we abandon trying to find an answer for these other folks who have, who were just doing amazing on our drug? And so that was, to be honest with you, the fuel. One of the big fuels. The other part of it, again, admittedly, selfishly, I don't know that I could have lived the next 20, 30, whatever number of years of life I had left, never knowing whether this was a true signal or not. Mm-hmm. All we wanted was an answer. If this was definitely caused by seladelpar, the safety signal, then that's fine. Then we did the right thing and we could move on. And then I could go find another asset or company to move on to. But we just wanted to find an answer one way or another. And so, to your point, one of the first things we knew we had to do is have a group of liver pathologists who would be willing to read the liver biopsies, both the screening and now the 52-week biopsies, scrambled and blinded. That was easily the first step. And then we had to assemble, of course, a broader panel of hepatologists that were expert in drug-induced liver injury. And I'll tell you one quick anecdote. A number of thought leaders told us you need one of these 3 pathologists involved. They're the top 3 in clinical development in Nash MASH. You need one of these 3 DILI experts in the country. These are the top 3. I told my chief scientific officer, I want all of these people. I don't want just one of mm-hmm. each of these pathologists and DILI experts. And we ended up getting all 6.
Oh, wow.
The top 3 DILI experts, arguably in the country, and the top 3 Nash MASH liver pathologists in the country involved in the analysis, because we had to look at the biopsies, concomitant medications, all the liver biomarkers. We collected a host of additional biomarkers for patients, and did what not just I believe, but Paul Watkins from the University of North Carolina, who was one of the DILI experts and actually the chair of the independent panel review, said himself, unscripted from CymaBay, by the way, when he presented this later at a medical meeting, the most thorough DILI investigation he'd ever been involved with in his 30-year plus career. This is what we set out to do, because we owed it to the patients to at least find an answer, and even to ourselves, to everybody inside the company, without question. And so we initiated this process. And the first part of that was to have the liver biopsies read. Now, not just for the typical Nash histology, but for everything that you might put under the bucket of these atypical findings, atypical histology that were flagged by the study pathologists.
And Sujal, when you were going through this process, I did hear some anecdotes that you were carrying tissue samples yourself in briefcases. I'm curious, is that true? And can you help me understand, if so, why'd you do it?
Yeah, look, so now think of the time frame. This is early 2020. We had already laid off 2/3 of the company. And we spoke earlier about rolling up your sleeves and doing everything possible whatever you need to do. And that's the situation I think all of us found ourselves in. One of the things we needed to do quickly was get all of these glass slides of liver biopsies imaged, in order to have 3 different pathologists reading the same biopsies at the same time as quickly as possible. Because now, again, remember, there were people trying to liquidate the company, these activist investors. So the clock was ticking. We didn't have months to figure this out. We had weeks, and at most several weeks. And so we needed to get these slides imaged so that there was high resolution images we could share with 2 of the pathologists, and then one would get the actual glass slides, right? This was just another task that came up. And it was critical that we do this as quickly as possible. Well, typically this is not an insurmountable problem, but it could take 2 to 3 weeks to get a contract in place, ship out your slides, get them imaged from some third-party provider. Money was not the issue. Remember, we had $200 million of cash on the balance sheet. We could pay $20,000, $30,000 to get these slides imaged, but it was time. And we didn't have the benefit of time to do this outsourced and take 2 to 3 weeks. So, I sat and I thought, okay, how do we do this quickly? Well, it just so happened one of those first companies I ever worked at after my graduate degree was an informatics company in Pittsburgh, Pennsylvania, that actually did a lot of high throughput imaging of glass slides. One of my colleagues at that company went off to work for Leica, the company that now makes the well-known microscopy company, imaging optics company. They make the number one selling high throughput, microscopy slide scanner in the world. So I called up my friend Scott and I said, and luckily I kept in touch with Scott, even though I hadn't worked at this company for 15 years. I saw him once a year, remained in touch with him and his family. So when I called him, I said, Scott, I need to use one of your scanners. He said, well, you could send it to our San Diego facility, but again, that's going to take 2 or 3 weeks. I said, no, I don't have that kind of time, Scott. The company may not even be around by then. He said, well, Rajiv at the University of Pittsburgh Medical Center has 2 of my scanners. And who was Rajiv, you might ask? He was a consultant to this informatics company that I worked at in 1999 to 2002. And luckily, I kept in touch with Rajiv since 2002. So this is almost 20 years later. I didn't keep in touch with him as much as I kept in touch with Scott, but I certainly kept in touch with him. And I called up Rajiv, who was the head of pathology at UPMC Shadyside. And I said, Rajiv, can I come in and use your scanners? I need to get 300 glass slides imaged immediately in a day. He's like, oh, of course you can do that, but at UPMC everything is requisitioned. You're going to need a contract. So I said, oh gosh, okay, so let's whip up a contract. This is not about the money. We're going to pay you, no problem. Whips up a contract. 20 minutes later, I sign it. He says, okay, now it's got to go to the CFO's desk at UPMC, and I don't know the CFO there. I don't know how long this could take days. It could take a week. No idea. So once again, this is like a storyline of problem after problem and just trying to find a way to solve it. Okay. And so now I'm thinking, how do I get the CFO of UPMC in Pittsburgh to sign this contract? I don't know the person. Well, I was involved on the alumni advisory board at Carnegie Mellon's Business School. This was a voluntary thing I had done for a number of years. It just so happened one year I overlapped with the treasurer of UPMC, who was an alum of Carnegie Mellon's Business School. And I didn't know him that well, but I knew him. I met him a couple of times. And so I got his phone number and email address, reached out to him, and I said, Tal, I have this contract in front of your CFO. I need it signed immediately. He said, is this paid? I said, yes, it's paid. Don't worry, you're going to get paid. And an hour later, he got it signed by the CFO. The next morning, I showed up in Pittsburgh where I had the glass slides flown in from Duke to begin helping the technician at UPMC put these slides into the scanner, image them. And the story that you referred to of me carrying the slides back, I was not going to check in these precious glass slides. So I put them in my carry-on suitcase and came back home from Pittsburgh to the Bay Area with the images in hand in a hard drive and the glass slides themselves, so we could get these 3 pathologists starting their review.
I think it's so important to highlight that the lengths you can go through for a patient to make sure we get these answers. And so I think concurrent to this, I want to highlight a few titles from some sell side notes, because I feel like it's not as well understood that when you're fighting the activist investor, you're trying to fight the external community and explain what's going on and trying to get answers for patients. You had a lot of your analysts who covered the stock said interesting things at this time, and I think this is worth highlighting. Quote, seladelpar is probably done. Move to neutral. Here's another one. Unclear future for seladelpar, but cash suggests potential upside. Downgrade to neutral. Another, downgrade to inline. Another roadblock for seladelpar. And the last one, all trials of seladelpar terminated or halted due to histologic findings, moving to hold rating. And so I can only imagine what it was like to go through all that and try to get these answers. So Sujal, one of the things we talked about was the esteemed panel. And before we get into, I think there was a biomarker analysis and the panel findings. I was curious, you mentioned at some point a letter. and how that was impactful to you. Can you explain that?
Yeah, I think throughout this conversation, we've talked a bit about what kept all of us in the company just striving towards at least finding an answer around what occurred in the Nash study. And there were a number of outreaches from patients themselves that, without question, were fuel to that motivation, not giving up, trying at least to find an answer. And again, as I mentioned, if that answer was seladelpar caused harm, I think we could live with shutting everything down and moving to the next opportunity. But in the moments where we didn't know, and there were so many forces against us to even find an answer, there were these patient outreaches. There was a patient whose family I met who told me quite literally seladelpar had not just changed the course of her potential life, but their entire family's life, their 3 children, husband, thinking they'd never need to see, his wife, their mother, have a liver transplant. And they were devastated that she had to come off treatment. She'd been on it for 2 years, all the way into an open label extension. There was a letter I got on Christmas Eve. As you mentioned, these findings we were alerted to just before Thanksgiving, before Christmas in 2019, before the holidays. We had to lay off 2/3 of our employees. And I think all of us were quite devastated at the time. And certainly I was as well, largely a shell, I think, of who I had been. And I was just astonished when on Christmas Eve, I got a letter from one of the leadership members of a patient advocacy group. Over the years, I attended a number of patient advocacy meetings, rarely ever seeing, CEOs of the companies that are represented there at those meetings. But for me, this was an opportunity to understand what it is people were living with that had PBC, and had developed just amazing relationships with the advocacy community. And so on Christmas Eve, when I got a letter from one of them saying, you all at CymaBay have been thinking about us and treatment alternatives for people suffering from these chronic liver diseases, just wanted to shoot you over a note and let you know we're now thinking of you and here to support you in getting through a very difficult time. And I was just floored to read that on Christmas Eve, just thinking she was thinking of us at that moment.
Wow.
It was pretty emotional, right? And these are the things that keep us going all the time. There's no question this is a relationship. We all in this industry, I think, truly appreciate when people get involved in participating in clinical studies. Those like advocacy groups that support broader communities of people suffering from disease. We just all want to be some part of that. And to develop a true relationship, I think, is really extraordinary. And it was something that I was incredibly proud of our entire team at CymaBay for years.
It's so fascinating, Sujal, because I think of it almost as a tension. You had the world-crushing pressure. I read the banking titles of all the analysts who were essentially walking away from your stock. There was so much skepticism regarding seladelpar and where the company was heading. The layoffs. We're on the cusp of COVID at this point, if I'm not mistaken. Things were just a very difficult environment, but you had this support, and so I think we're now at the stage where I believe the data start to line up, that maybe things are not as we thought. So I believe, if I have the sequence right, there was a biomarker analysis, and then the esteemed panel shared their findings. Can you start to walk us through how all that came together?
Yeah, and a reminder, one of the key elements of the independent panel review was a reread of all of the liver biopsies in the 52-week Nash study. So what we did is we took all of the screening biopsies before these patients ever took a dose of seladelpar, and we scrambled them and blinded them with all of the 52-week end of treatment biopsies. Some patients didn't finish the 52 weeks, but most actually did. So we took all of the end of treatment biopsies effectively and the screening biopsies and scrambled them and blinded 3 additional very experienced hepatopathologists. We asked them not only to score the elements of Nash, those that are part of the NAFLD activity score, steatosis, lobular inflammation, hepatocellular ballooning, but also to score fibrosis, in that setting, but also all of these, quote unquote, atypical findings that the study pathologist observed at the end of treatment biopsies. And so, this was a process whereby we didn't know how long it would take them. They all have their regular day jobs. We talked a little bit about the rush to getting the biopsy slides imaged so that we could get all three pathologists reviewing at the same time. But it was an uncertainty as to how long it would take and whether we would have enough time, before many of these activists tried to force liquidation of the company. But we steadfastly continued to dialogue with those independent pathologists. They conducted their blinded review. And before the full panel met, that included some of the hepatology DILI experts, before the full panel met, we actually had, at the end of the pathology review, a subcommittee meeting with those three hepatopathologists, myself, our chief scientific officer, and our head of regulatory, just to listen to and hear their findings. They remained blinded as to what they were observing, but we got on a call at some point to hear their review.
Walk us through all that. Was it a singular moment? I'm just trying to understand. I've always been curious about how these panels share their findings and what that felt like.
Yeah. In all honesty, we were getting at least some of the reads back before they had fully completed their review. So they remained blinded. Of course, on our side at the company, we were unblinded. We knew which slides they were reviewing, whether they were pre-treatment or post-treatment. And I could tell you that early on, we were already getting a signal that some of this atypical, unusual pathology was actually showing up in the screening biopsies.
It was there already.
But even then, Kenny, here's the distinction. Even as we were seeing some of those signals, we knew that the only way for us to proceed was to maintain the blind, maintain the integrity of their analysis. Remember, the goal for us was not just figuring out a way to get seladelpar back to patients. It was just finding the right answer. Mm-hmm. And we didn't want to do anything to interrupt the process of finding that right answer. And so we were incredibly disciplined at not sharing any of these intermediate results. But I could start to sense that there was some of this at baseline, which made some sense, right? Because I mentioned there weren't any elevations in typical liver enzymes associated with liver inflammation or liver injury. There weren't clinical events or clinical symptoms. And so to see some of this in the early reads, we were encouraged, but we knew it wasn't over because there could always still be an interpretation by these independent hepatopathologists that was aligned with the interpretation of the study pathologists, that in fact, these are somehow maybe getting worse over time or inconclusive. We always knew that one of the results of this independent review wasn't just the two outcomes of seladelpar definitely caused DILI or definitely did not. In fact, we would all argue that the majority probability outcome, the greatest probability outcome, was it would be unknown, that maybe there was some suspicion, maybe it did something, maybe it didn't, which for all intents and purposes in drug development means the program is still over.
Correct.
And so we were incredibly cautious about getting all that excited and emotional. We were, again, encouraged by it. It emboldened us to make sure that we finish this review and have these independent panelists finish their review with the plan that we actually outlined for each of them and that we actually shared with regulators. And so that's ultimately what we did is wait until all the way at the end, we had an opportunity to hear their interpretation of their findings.
And do I have this right, Sujal, that it basically what was established at baseline in these patients was not acknowledged in the analysis, and the panel helped uncover this, that these findings were there already, that seladelpar did not cause this issue, it did not exacerbate this issue, and it frankly, is it appropriate to say human error?
Yeah, I might not call it human error, but the first things that you mentioned were absolutely true, that in the end, when the study pathologists completed their review, there were in fact two things that they highlighted to us. And you asked about painting the picture of was there this singular moment. There effectively was a singular moment. And I recall, being at home, because now we're quarantined in the midst of the pandemic. This is, now early 2020 when everyone in the world was sent home. I remember walking to my backyard, getting ready for this call, notebook in hand, ready to jot down all of the slide numbers that they were concerned about being suspected of drug-induced liver injury. And I always imagined, they would highlight some of the slides that the study pathologist highlighted. They might highlight some that the study pathologist did not. Maybe they'd miss some of them that the study pathologist flagged as well, right? But largely, I was ready and prepared to write down the specific slide numbers that were concerning to them. The first thing they told us, in fact, was that they saw these features of histology that the study pathologist flagged and called out. But even before they were unblinded, they suggested to us that none of what they saw appeared to be suggestive of drug-induced liver injury. And my jaw almost dropped at that moment. And of course, now you have a situation where even the interpretation itself was different than these other two study pathologists, who in fact were their colleagues. They are all part of the same societies, all distinguished in terms of their experience, and yet their interpretation of what they saw was quite different than the interpretation of those two other pathologists. So now, I don't call it human error. Let's just call it being human itself. I think because we all, as human, can be fallible. We can all miss things. And so I don't think there was anything relative to intent at the time. But it just so happens that, when you're not looking for something, you're not going to find it. But when you start to look for something, you may very well find it. And I think that's largely what this case was. There was probably an end of treatment biopsy that had some of this unusual pathology, and then the study pathologists, human minds, become even more focused on looking for more of that, and intensely focused on it to the point in which I think they largely convinced themselves they've never seen this in this disease. But in fact, in these patients whereby they flagged the end of treatment atypical histology, all these patients had that atypical histology at baseline. And it is true that in the field of Nash MASH, this had not been previously widely published. This is not something that, when our situation occurred, we could just jump into the published literature and say, oh, you know what? There's all these findings that are consistent with what we're seeing. It just so happens that while this may not be part of Nash MASH etiology, some patients with more complex inflammation may very well have interface, or some of these other features that were flagged that had not been in the published domain. And so we ultimately were one of the first groups to at least report on it in the fashion that we did. And I will say, subsequent to the entire panel's findings being presented at medical congresses, other groups went back to their Nash MASH liver biopsies, looking specifically for the things that were flagged in our study, and sure enough, later published that they saw some of those same pathologies in this disease, not in all patients, but in somewhere between 20 to 30% of biopsies, for example, in one paper published, by a thought leader in the field. So this was something that, sadly, we had to be the first to identify, but luckily we did. The entirety of the panel then reviewed everything collected in the study, not just the pathology and the biopsy readings, but also biomarker analyses, DILI analyses. All of these things were done in incredible detail over several days, looking at every single case that the study pathologist had flagged. And it was only through that incredibly thorough review that we felt satisfied for the panel to then render its opinion. Again, this is independent of the company, and the panel themselves adjudicated every one of these cases and ultimately unanimously determined, and this was a statement that they put out that was printed, shared with regulators, again, eventually presented at medical congresses, their unanimous decision that seladelpar did not have any signal of drug-induced liver injury, either by biomarker or clinical events or symptoms, but also not by pathology. And that's ultimately what we knew it would take, again, not just to convince investors and analysts, but more importantly, to convince patients that we would enroll in future studies, and of course regulators as well.
So I want to pull on a couple threads here. So one thread is, doing this reanalysis of the liver biopsy slides, because it's the right thing to do to figure out, what's going on here. Are we actually harming patients? Are we not? Underneath that is still, given, the first round of results, how, A plus B doesn't equal C. There's some piece of the puzzle missing that probably gives you it retains a lot of conviction that this is a drug that's both safe and effective, and we should figure out, the truth around this. And underneath that, though, is there's this, obviously this process takes a lot of time. And, you're, as the CEO of the company, tasked with like preaching patience, right? To not only, the wolves at the gate that are trying to, take over your company and turn it into a chop shop and, rip it up for parts, right? But, your board, other potential investors, your colleagues on your executive team, all the folks working at the company. So, take us through kind of as a leader, like what your strategy was there. How do you get those folks on board? A lot of times, obviously, your board and everybody has to be on board with this plan, but they probably get calls every week. Hey, why aren't you guys turning this company down? What's your CEO doing? What every time that call happens, they probably are second guessing themselves a little bit, being like, man, should we just talk to Sujal and just, pull the plug on this type of thing? Like, how do you, it shows, you have a ton of conviction in your strategy, a ton of backbone in saying, hey, this is the right path forward. How do you preach patience to, all your stakeholders, to get to the finish line on something you had deep conviction about?
Yeah, it's, Shivu, it's really insightful. I will tell you that the forces against us, not in a malicious way, even in some cases, but simply inertia. One of the things that I learned in this process is, when you go through something like we did and it's seemingly game over, the easiest thing for the world to do is to say, thank you for playing, and they move on. Having relevance, getting people to care, whether those are experts you need involved in the independent panel, your own board members that you need to help, allow you the freedom to move forward with what you're trying to accomplish. Forces to hold off, the external, activists, for example, that are just trying to make a return on their investment, post the downfall and forced liquidation. Forces that include searching for other strategic alternatives for the company. We had $200 million of cash that could easily be merged into some other company and admirably be used to advance other assets, that might help other patients. The number of forces and the force itself was extraordinary. And I can't tell you that I reflected at the time what kept us moving forward. I can tell you that it required being incredibly relentless. I can tell you that it required, the team coming together without question. There are times when I've been asked about these series of events over and over again since they happened 5 years ago, and people ask the same kind of question. What have kept you going? And I think a lot about when you listen to people in the military that have been in conflict, those that have not, are admirable about their fight for freedom or liberty or justice, whatever it may be. And when you interview that soldier, if you will, all too often you hear them say, well, of course I'm for freedom and liberty and justice, but, I was fighting for the person next to me. That's all I thought about. I thought about the life of the person in the trenches next to me. And I think that was part of our mentality, at the company is, those of us that are still there, we're there together. We're going to get through this process together, and either, the company survives or it doesn't. But one way or another, we're going to complete it together, and we're going to fend off the forces that are otherwise just trying to to have us fold our cards, if you will. I think a lot of it was just going in day to day and fighting for the rest of the team, just to keep it going. It wasn't probably much more than that. And for sure, the inbounds from patients and what we knew about the potential for seladelpar, particularly for patients with primary biliary cholangitis, that was absolutely the fuel to keep it moving. But so much of it was just banding together as a team, I would say.
Sujal, I'm curious. We didn't talk about this, haven't asked it yet. I remember you shared you were approached by one of the activist investors, I think, in a parking lot when you were on a way to a meeting. And again, it just the environment being so difficult before things cleared. We never asked about your health. Was there ever a time where you thought, I have to take a step back, get a little refresher, step away for a little bit, maybe take a weekend somewhere? Were there things you had to do for yourself, or was that not a consideration throughout all this?
I don't really recall it being. I think maybe I was just fortunate to be healthy enough through all of it. The mental part of it though was again tough, but those are the things we fight through. Many of us just to do the work that's in front of us, right? It's a little bit of ignoring and pushing it off. The level of stress and anxiety, particularly raised by the activists, because there was one lead activist that owned 10% of the company that had gone public. He'd effectively hired attorneys, gone public with his dissent of management, criticism of management, effectively wasting shareholder capital. And what ends up happening is you have a half a dozen other amateur activists who realize they don't have to spend any money on lawyers. They can just buy a little bit of stock and then call me every day to pressure me to liquidate the company. And so it wasn't just one activist. That's part of what people see on the outside. It was about a half a dozen of these folks. And, I can only smile now because we survived it. But I like to tell the story that I met every flavor of activists. There's, the standard aggressive ones that get on the phone and berate you and threaten your career, threaten the rest of your financial security for the rest of your life. There's passive aggressive activists who gets on the phone and says, Sujal, this was never your fault. You're young. You're going to find something else to do that's going to be amazing. I'm sure you're going to do the right thing here and just shut down the company, stop, spending money and just return cash to shareholders. And then there was even good cop, bad cop activists where one would get on the phone and berate me for 10 minutes. His or her partner would jump on the phone and say, well, maybe Sujal is doing the right thing by doing this investigation. And they just go back and forth. It was mind games, quite honestly, mind games. And, Kenny, you mentioned somebody, surprising me in a parking lot as I was headed to a cafe. And, this was an investor who came up as a friendly, hey, I'm an investor in CymaBay and I have all this experience on boards. I think you should consider adding me to your board. I can help in this activist type of situation. Turns out it was an activist themselves, trying to disguise wanting to be helpful and friendly. And an activist show up at an investor meeting uninvited. And so, I can't tell you what it took other than, just a lot of grit and perseverance, and just trying to keep in mind why we were doing this in the first place.
Okay. And so my understanding is with the sequence of timing, we're heading into COVID, the stock was right around $1 or $2. The panel clears seladelpar, and it was a pretty meteoric run in a matter of weeks. I believe the stock returned to somewhere around $8. So, maybe 6 to 8 times higher from where it was. Is that what closed the door on the activist pressure and some of Wall Street skepticism?
Yeah, absolutely. Look, I think, by the way, one quick backtrack here around the emotional element. When, the small team of us who were leading this independent review from within the company with the independent panel, obviously, everyone dialing in remote, when that final fourth day of adjudication concluded and the panel had typed out their unanimous finding, I did then go back to my office. A few of us actually gathered in the office with our masks, but to do these adjudication meetings for over 4 days. And then all the motion came out. So it was pent up for months. And that moment when I realized we finally made it through and it was never seladelpar, that's when, I had my private moment and all that emotion came out. We then, of course, as you were alluding to, shared these findings publicly with the street. The stock tripled, and frankly, the activists made more money, much more money, from the outcome than they would have in liquidation. So they were quite happy. I didn't hear from most of them. The lead activist did call me, and I can only say this after having reflected quite a bit on it. We never really had overly contentious discussions. I mean, they were tough discussions. I felt like I was on cross-examination with him every day, and I had to watch every word that I was saying, and I had to be careful that I didn't add fuel to the fire for him. I couldn't just be angry. I couldn't mistreat him. I couldn't ignore his calls. I had to take every one of his calls, which is pretty much every other day for 4 months.
Wow.
But I did my best to treat him with respect. He was doing what he felt was right for him. I'm glad that's not the path I've chosen to make money. But when we made the public announcements, he called me and he had gratitude. He said, "Thank God you stuck to your guns.
Wow.
You're the right person to be leading the company. Your team is the right team to be running the company. And I'm going to be pulling my proxy." He had filed a proxy to nominate his own slate of directors. And so when I talk a little bit about timing, we finished this independent panel review 1 month before our annual shareholder meeting, where we would have had to go up against his slate of nominees. And you can only presume that he would have nominated directors that would have voted for liquidation. So all that happened just about a month before the annual meeting. But he was quite pleased because, again, they made a lot more money with the outcome at hand than he would have in liquidation. So, but I will say that from that point forward, he'd always been respectful. And, even leading all the way to the point where the Gilead announced acquiring CymaBay. We had announced the agreement to be acquired by Gilead. He even sent me an email that day, just sort of a chuckle, but a congrats, congratulatory email.
Well, it's such a fascinating turnaround, and so I'm kind of skipping through some time now, and it simplifies what you went through. But the stock went up, and then through 21 and 22, I noticed it regressed a little bit. And one of the things I wanted to ask you was, did you still face skepticism around the drug? And so before you share, I wanted to highlight something. I don't think I've ever shared this with you before. We had a lot of hepatologists. We had a lot of esteemed clinicians, frankly, at Gilead. And I wanted to share this with you. I had a private conversation with one of them. Brilliant guy. I've learned a lot from him over the years. But I remember after I closed the door and I said, "It's been cleared. I think it's good to go." He said, "No, there's something wrong with that drug." And he reminded us of the old dosing issue. Remember, I had to dose lower for PBC, all this stuff. He said, "There's always something with that drug." And I said, I the panel disagrees. And so when I was looking at, the share movement and the story and how it kind of pulled back before you started to present at AASLD 2022, that's Sujal where things took off and never looked back again. The meteoric move. Were you still facing skepticism? Were you still having to convince people?
Largely not, Kenny. I would tell you that, folks were quite pleased with how thorough the independent review was. And including regulators. I mean, regulators took less than 3 weeks when we filed our complete response to the formal clinical holds to lift those clinical holds. Regulators, the agency, the FDA, for example, just told us they couldn't assemble a panel that was as esteemed as we had assembled in our independent review, even for an advisory group. And so they were quite pleased at the process, the individuals involved, how we went forth at finding these conclusions. Largely, even the external community was quite supportive. However, to your point, there are always individuals who remain skeptical. And let me mention something that I think is really important in our industry, in drug development, right? The efficacy and safety of a drug is monitored for the life of a drug, not just up until approval. And so, safety is something that we, if we were still independent and commercializing seladelpar, of course, now Gilead is always assessing. So there's no end to that review and collection of data. So, I think it's important for those of us in drug development never to be overly conclusive about anything that we see, because that's our responsibility, right? It's our responsibility to continue to monitor and continue to. But I have to be quite frank, most people accepted it. The dip in the share price, I think, had a lot after the rise initially after the announcement. I think had more to do with the fact that, okay, investors realize this company is going to need to raise more money to fund now and to enroll a new phase 3 study. So one of the challenges for us was, if you recall, the phase 3 registration study was fully enrolled, but we had to stop that study and eventually terminate it. We couldn't keep patients on drug holiday for 4 months and maintain the integrity of that study. So we always knew we would have to, at some point, re-enroll another study in, again, what is a rare disease. These studies take many months to fully enroll. And so I think part of the challenge had become, the investors realizing we're going to need to raise more money. It's going to take over a year to enroll a new study and then another year to get to data. And so there's this long period of time before which anyone would see the actual set of phase 3 registration data. And I think that was part of the initial dip, after the announcement that seladelpar had really been cleared. To your point, though, again, there were times I faced it. Let me focus on one other group that you mentioned, the analysts. You mentioned some of these analyst quotes. I don't fault any of them, right? I don't fault any of them for those quotes. So that's the information they have in front of us. It's the information we had in front of us, right? So at that moment to say seladelpar may be done, I don't fault them for that. That's the environment. That's the situation we were frankly in. And most, of course, were again very complimentary of the process and the independent review and the depths to that review that we had gone through. Again, we had presented that at medical meetings, as you know, in our industry. That's the hallmark of having true peer-reviewed data, and I think that did a lot to lift any potential concerns. But remember, we're all human, and so the individual you spoke to, there was even an analyst, I will tell you, that covered CymaBay at the time, who was considered the top research analyst on the street. Even to this day, he is. And he's at a different firm now. I won't mention his name, who was incredulous. He got on the phone with me and said, "So you're telling me, Sujal, they just missed it?" And I said to him, sometimes the easy answer is the answer. And as he was just incredulous of how could this happen? And I told him, I said, because we're human. Humans can make these mistakes. And the simple answer was the answer, that this was just missed at the screening biopsies and nobody had been ever looking for it. And so, I think that's just another lesson of mine is sometimes I used to say this inside of CymaBay. I'll tell you it now because I think it's actually true. One of the reasons I think we survived is because I wasn't the smartest person in the room, to be quite honest, because most of the people that were far smarter than me said it's over. You're never going to figure this out. You can try and it'll take 2, 3, 4 years and you don't have that time for the life of drug development. And I think part of the reason we survived is, I wasn't the smartest person in the room, didn't outsmart myself into thinking that it's impossible, frankly. And so I can only imagine the person you spoke to that had skepticism probably knew liver disease better than I did. And as a scientist, maybe a clinician scientist, a lot more experience than I did. And sometimes that can, put us in a difficult situation as well. And so I think at the end of the day, most were really supportive. And I don't think there was a massive cloud over us, but there were always individuals that remained skeptical.
I think that's also a great example of why, or two things. One, all of us on a regular basis deal with people who have expertise in particular areas. And that's why you have a team, right? It's multiple perspectives. Which angle are you seeing that someone else is seeing that another person might not be? And, these decisions aren't always made in isolation, right? It's considering all the information and both also kind of all the strategic paths that you laid out. I think the other thing that stands out to me too is that, or a reminder, I think, for folks is that, anytime for most of these drugs that, we're trying to develop, we're on the cutting edge of science and we run into stuff that has never been run into before, right? The problems that we face are new and different. And, maybe to some extent we have analogs we can lean on that will help us solve these issues. But, in the case of the liver biopsies, right, as you were saying, this was a new issue that even the experts had not encountered before because they weren't looking at it. And, now that it was elucidated, they could go back and really have a fair assessment of what these patients were experiencing.
I think it's absolutely true.
And Sujal, that's where things really started to change. You presented amazing data, AASLD 2022. That's the premier liver conference for our industry for liver. And I remember, I think there was improvement in lipid parameters and a few other measures. I think we were already aware of the pruritic benefit as well, the better safety profile. So now that we've gone through and seen that the drug is not to blame for any of this, we had that presentation. And then I think a competitor read out data. Now we're heading into 2023. Competitor read out data where they achieved their primary endpoint, but they didn't have that pruritic benefit. So in a competitive positioning, it seemed like seladelpar started to edge forward a little bit because I think that competitor was one that was watched. And then, of course, you had your positive phase 3 data. And then shortly thereafter, Gilead. We're going to post this image because it's so impactful. It really is very much a rocket ship move. And so it's almost like all the things that were so painful and challenging, they were all washed away. And then it seemed to be like things just worked out for seladelpar and CymaBay by staying true to the science, staying true to the patients. Things almost locked in place quite perfectly. And then you had an exit with a partner who has rich legacy in liver and can really drive this forward globally.
Look, I think we all in this industry will mention certain things, and it's just a matter of who actually behaves that way. So we talk a lot about following the science. And I think many in our industry do, of course. It's not to suggest, we're in a minority for having been incredibly true to what the data is telling us. But there are instances where some people are not. But largely, that is what drives us in this industry. And, it just so happened that the initial phase 3 study that we terminated early, we had a fairly significant number of patients that had at least completed 3 months of treatment. This was a 12-month treatment study, like registration studies are in PBC, but we had at least a majority of patients finish 3 months of treatment. And so, although only a couple had actually finished the full 12 months, many had finished 3 months. So we modified that statistical analysis plan before unblinding that study. And that's a method to maintain the integrity, of that analysis by keeping it blinded while you modify the statistical analysis plan to say, now, rather than looking at all of the endpoints at 12 months, we're going to review them at 3 months. And so you set that ahead of time before you unblind. When we did that with that initial phase 3 study, this is some of the data you were talking about in 2022 that was presented and eventually published, we hit the primary endpoint with high degree of statistical significance at just 3 months. And that was not a surprise. In fact, vast majority of the effects of the drug, the kinetics of this drug, are such that you see almost the maximal effect at 3 to 4 months on the liver enzymes associated with cholestasis that are part of the approvable endpoint. And so we hit that primary endpoint with high statistical significance at 3 months. In fact, we hit both of the key secondary endpoints, one of which, in fact, to your point, was pruritus, this effect on lowering itch in patients with PBC, something they suffer from, and that leads to a decrease in quality of life significantly for people living with PBC. We hit pruritus with statistical significance in just 3 months. And that was, in fact, I think something we had not yet measured before in prior studies. The biomarker was not surprising. The itch was pleasantly surprising, although, again, we understood the mechanism and it followed the biology. It followed the science of what we should see is what we ultimately saw in that study. So when we committed to a new phase 3 study, we were quite confident, certainly encouraged by the prior results we had seen, that we would once again, see positive data on the primary endpoint, and fingers crossed we'd see the same consistent effects on itch. And when you line up that full phase 3 12-month data next to the 3-month phase 3 data from the study that was terminated early, the data is almost identical. And so that's another thing that, when you're talking about skeptics, to see very consistent, both efficacy and safety out of 2 separate now global phase 3 studies, that's where, our confidence comes from relative to, again, just the risk benefit. No one's saying that you're not supposed to be following safety for the life of drug, but the risk benefit, I think, was such that there was a real opportunity to improve lives of people living with PBC, slow progression of disease potentially, and also reduce symptom burden. And that's the profile that we had in hand. And I think much of that over time led to the rise you were referring to relative to the share price and the value of the company. And there were other decisions along the way. As we became more and more confident that we would see positive phase 3 data at the company, we had a lifecycle management team commit to an additional study that would help potentially one day support broadening the patient population, those patients that could actually benefit. And that was another key element of seeing value rise, because I think the external world, investors, but even the medical community began to recognize that this drug could be used potentially, if that data is also supportive, in a much broader population. I think that's some of the excitement that continues to exist today, even in the program, while it's under Gilead's hands.
I think that's so interesting. It's almost like, because of the downs that you had to dig yourself out of, the process of going through that, the process of having to close your initial phase 3 study, start a new one, coming out the other side of it, because of all the resilience and, all the things that you had to do to build that evidence package, you now had this really robust evidence package that showed a much more clear signal that this was a very safe, efficacious, and approvable drug.
For sure. Shivu, you're always trying to find silver linings when you have to. And although, had these unfortunate events in the Nash study not occurred, seladelpar might have made it to patients sooner. That's the past. We can't focus on that. Have to reflect on the here and now and in the future. And I think you're absolutely right. The silver lining was we had amassed even more evidence. We had amassed even more safety data. So that's important. We care about these things. It's not about disagreeing with skeptics. It's about actually generating as much evidence as we can to give patients comfort, to give physicians the comfort to prescribe the drug. And so the silver lining, yes, was we had even more data than you might otherwise have needed to get the drug approved, and that's a good thing. That's a good thing when you have more efficacy evidence and, again, more safety experience.
And Sujal, CymaBay is not going to get any credit for this, but part of the task of the industry, of what we do when we publish, when we try to advance the clinical and scientific boundary, is to also advance how we look at things. And that's something that if the drug didn't have these issues from the Nash study, it probably wouldn't have been established that, maybe we should look at these samples at baseline. So there were things that I think CymaBay painfully charged forward through, and the whole industry benefited. Patients definitely will benefit. Clinicians will definitely benefit. So I feel like there's even process. It's not the most seductive thing we think about in our industry, but there was a process benefit as well that will never be remembered, but I think is so important, because now liver is going to look, and maybe even other therapeutic areas, that maybe we should look at things a priori.
Yeah, I think so. I think certainly those in the NASH/MASH field will be, post our circumstances, encouraged to read and assess baseline biopsies with much more detail than otherwise we had been doing in the field before. Again, these things are all to the benefit of patients at the end of the day. The more we understand about the disease, about the effects of treatments, being studied for these diseases, in the end, I certainly think patients benefit. The field benefits, to your point, Kenny, and that's an extension. And as an extension, ultimately, hopefully, the patients benefit as well. And so there's no question. I mean, we had lessons learned ourselves, things we should have and could have done better. And luckily now, important. We felt it was always important for us to share these things in the medical community, so that, as you point out, they could help others think about the best way to design studies and to move other programs forward.
Yeah. So just on the timeline, Feb 24, Gilead acquires CymaBay. And I remember we got lunch shortly after. And I asked you, it must feel so good. And I was kind of piecing together the CymaBay story. And I remember you shrugged it off. We had that nice lunch. You said, well, the drug's not approved yet. And I just remember thinking how singular and laser focused you and your team were. Even with, a lot of companies seek the buyout in across all industries. It's not just healthcare. And even then, in your mind, job not yet done. And so I think you were still involved with the drug. I think you were still in an advisory position with Gilead. And then, of course, the drug gets approved. It's such an amazing story. It's so touching. And so now that we're getting to the other side and things have changed now, one of the first questions I wanted to ask you was, you think back, I think I know the answer. Would you do all this again?
No question. I'm not one to reflect too much on the past, so I don't know how I would not have done it again. It's to get to the finish line was incredibly gratifying. I think when you and I had lunch, it was, I got to mention this quote because I just think it was an amazing one from Kobe Bryant, before he had sadly passed. And I can't remember which series it was. They were up 2-0, maybe even 3-0. I think they were up 2-0. And he was in the post-game press conference, and he looked serious. There was not a smile on his face. And some reporter said, "What's the matter, Kobe? You're not happy? You guys are up 2-0. Why are you not smiling? I thought you'd be a little happier." He's like, "Job's not finished." He said to the reporter, "Job finished?" "I don't think so. Job's not finished, right? They hadn't finished. They hadn't actually won the championship yet, even though they were up 2-0 or 3-0. And that's how it felt for me. It's the outcome in many ways was bittersweet. We had been championing the effort for seladelpar for so long, and particularly given the struggles we went through. This was our baby, so to hand it off to someone else was not necessarily that easy. But let me just say, because you mentioned it, Kenny. Gilead is a tremendous company that has been incredibly successful in the broader liver field and in other areas, and I know that they have the opportunity to take seladelpar to more people around the world in a shorter period of time, perhaps, than we could have. And so, that's what keeps me smiling now, is knowing that seladelpar is in good hands with a very experienced group with I think broadly high integrity, and now seladelpar will improve the lives of likely more people around the world. And that's a really important takeaway. Having that feeling of it being bittersweet and taken away from us is momentary, only in so much as then I think about those patient advocates, not just in the US, but in Canada and in Europe, and knowing that seladelpar might get to them much more quickly. That's a big deal. And that matters. And so, we think, clearly there's the investor return element of M&A takeouts in our industry. But underlying those are the potential for these treatments to reach many more people. And that, I think, is really key, of course, to focus on. But there's no question for me, as you mentioned, in our industry, you see many companies taken out before drugs are approved. And in many cases, they actually never get to the approval line. They never have successful development programs in the end, even though the acquisition happens and investors make money. And so to be in a position where our investors and supporters make their return from the M&A, but importantly, the drug gets to approval, patients benefit, the broader medical community benefits, and of course, everybody inside of CymaBay and now inside of Gilead have the pride that they are part of a journey to bring a new treatment to people with PBC. That part shouldn't ever be discounted either. It's what keeps us going. It's what helps us think about the next one to work on, right? So,
Sujal, I think a lot about leadership, and when you run a publicly traded org, but you also work in our industry, we also serve patients too. And there's this push and pull between, getting the returns for shareholders versus doing what's right for the patient, because sometimes the timelines, they do not align. Do you think this formula is too simple, that if I take care of the patient, the shareholder will be happy?
I hope we all keep that as our North Star, because I do believe that works. Does it work every time? Perhaps not. But if you, again, back to follow the science, follow the data, keep the patients in mind as your North Star, and you will rarely go wrong. Now, there's a lot of other things that have to happen too. It's not so simple as that. That should still be the focus, but then what are the other things that matter? Honesty, transparency, those things matter quite a bit. I think one of the things I heard from many of our investors through thick and thin was that they felt we were more transparent than most companies they had invested in previously, or many companies they had invested in. I don't want to speak with hyperbole, but even when we had these unusual findings and we knew very little over a weekend, most of our investors said you shared more with us on that call Monday morning than we typically hear from others. Too many other people try to protect something by not sharing enough. And even though you're still exploring and even though you don't really understand the situation, we appreciated the level of transparency and the sharing of information. And I think that's a principle that sometimes is lost in our industry. And again, perhaps not maliciously, but sometimes just lost. People don't focus on the fact that, be honest and be transparent and forthcoming, and then manage the circumstances that may follow that transparency. In some cases, it may mean you have to work harder. It may mean you need to collect even more evidence to convince the next investor to participate. It may make the next financing harder. But if you're true to being transparent and honest, in the end, I think it always serves you well. And to your point, Kenny, all of that should just be surrounded by an ultimate focus on developing these treatment alternatives with the patient in mind.
I think it's a great point, and it highlights some of the, oftentimes some of the misalignment that can happen with, the incentives between what investors are looking for and kind of what people at the company are aiming to do. I guess related to that, if you take the full arc of CymaBay and let's transplant that same arc into kind of today's environment and today's climate, do you, maybe an impossible question, but would love your thoughts on it. Do you guys get to the point where you're, the rise of the Phoenix phase of the company? Is that, possible in today's climate? Or, I guess, how do you think about that, just given kind of where we are as a sector today?
I like to hope it's still possible. I think everyone working in this field has to believe that that's all still possible. In the end, I have to remind myself, as all of us do at CymaBay that have been part of this journey, that we had a really good drug. And, that's not easy, right? In our industry, what is it? Only one of every 10 programs succeeds, and roughly. And we were fortunate. We had a really good compound. We had a lot of amazing people dedicated towards the development path of this program, new ideas for development, just really diligent focus on executing the right program, following the data, following patients. And so there's so much you need to still make it happen. But we did have a good drug. And I like to think that if we in this industry continue to focus on the science, be honest, not just externally, but with ourselves, that we should be able to continue seeing these stories. Of course we can, right? Ours is not a singular, unique event. We've seen this in the industry many times, ups and downs, and ultimate success with drugs making it all the way to patients. So I certainly am a believer that it continues to be out there. And we see it every day. I think there are amazing people in our industry. I can tell you, while our story is unique, and I think there are elements to it that I'm incredibly proud of, most of those elements are the actual people surrounding the story itself. I think we see this all around our industry. And one of the things that I think is also amazing in our industry is we cheer for one another. In our moments where seladelpar had its ups and downs, some of my closest colleagues were running companies that were our direct competitors. And that's really unique, I think, in our industry. But it's always with this mindset that, hopefully we're successful, and then hopefully somebody else after us is more successful and can take the science even further, right? That's the idea. And so we support each other in ups and downs in a way that I think is quite unique in this industry. I certainly relied on many friends in biotech in those really tough moments, just to to continue to need that motivation to keep going. And now on the other side of it, hopefully I'm there for others in that same way. But I think those arcs are still out there.
Sujal, now that we've talked about how it resolved and how things have concluded with the Gilead acquisition, I think it's important for people to understand what's next for you. How are you thinking about your time now, and how will you think about maybe getting back into another leadership position, and what makes sense for you?
I need some time to continue to reflect and decompress from this journey. It was quite literally all of everything, I certainly had individually. So I've been taking some of that time. I can't tell you I've got a specific timeline to when I'd feel like jumping back in again. I still stay involved. I sit on 2 boards. It gives me the opportunity to help, other teams with their programs, which I love doing. I spend a lot of time just voluntarily advising people who reach out, just looking for advice or input or wanting to hear some parts of our story even. And that gives me a chance to still be involved. It's just supporting others in the industry. I think at the right time, I certainly would love to do it all over again. And I distinctly remember my last moments in the company, my last day at the company, in fact, telling the people that in the end, I think this is the case for many, not just myself, but in the end, we think most about the people that we were surrounded with and our teams. And for me, that was without question what would bring me back, is the potential to work with some amazing people again and really enjoy it. I used to once think that only athletes, actors, movie stars, rock stars could love their work. Like, who wouldn't love that work if that's what you had to do to make a living? And then somehow I found CymaBay, and I loved it. I loved going to the office every day. I loved doing the work. And so much of it was about the people. It was about the mission, but so much of it was about the people. I'd love to have the opportunity to do it again one day.
Shivu and I have asked you so many questions. Is there anything that you wanted to share that maybe we didn't get into, aspects that perhaps we didn't graze yet?
Boy, I think we covered a lot. I think one of I'll leave you with one other thought since you've asked the question. This is something that I've talked a little bit about, and something I learned just because of our own circumstances. One of our scientists at CymaBay gave me this great Michael Lewis book that I read during the time we were on clinical hold and we were uncertain about seladelpar's path. Is this book called The Undoing Project, and a book that chronicles the lives and work of Daniel Kinnerman and Amos Tversky, 2 Israeli psychologists that eventually won a Nobel Prize in economics. They were not economists, but just in terms of how people think, and upending what we all believed around decision theory, probability weighted based decisions, and things that I think maybe aren't always the case. I think the thing I reflect upon as I read that, and I think a lot about us as humans, is it's important for us to seek external input, expertise. Those things are always important, from our own teams, from experts in various fields. I think one of the things we all probably could benefit from more is just self-reflection, and just look inward more often than just looking for answers externally. And I think that will serve many of us well when we think about this industry and what it is we're doing and why we're doing it, is sometimes just to take moments to self-reflect and look a bit inward and trust yourself. That's, I think, something that served us well in the times where the external world, like I said, had already passed us by. We were just in the process where that's all we were left with was ourselves and looking inward. But I think it's something that I've learned, over time, and I tell others in tough situations in our industry, navigating things, is, seek the external input you need from board members, from experts, from colleagues, but take some time to look inward and self-reflect. And sometimes you'll find answers inside yourself, and trust it.
Sujal, as I think about, or as we conclude, if you remember, we started this conversation with how difficult it is in healthcare and how so many times you hear stories from patients where they feel like they're alone. And so that was initially how I thought of framing this, that CymaBay's story can be an example for how we can serve those patients. But as I heard you talk, I dare say it's bigger than that. We're at a time where fundamental leadership, truth, and honesty are in question. And so when I think about your story and what CymaBay was able to do, I think about it as really a candle in the dark. It's not just about healthcare. It's about leadership today. It's about being true today. And that seems to be in question in a lot of areas, in a lot of places right now. And so I'm so grateful to you for not only what you accomplished, but for also being available to share these things. Because I think there will be others who listen to this and say, maybe that's a good model for us. Maybe we should try that. And so the impact of what you did isn't just the approved drug, isn't just advancing the scientific and clinical boundary. The ripple effects continue. And that's what we're trying to contribute with this conversation with you. So thank you so much for joining this. It's been so eye-opening and enlightening to hear.
I appreciate it. Thank you for listening, and thank you for sharing the story.